welcome back to the Deep Dive. Our mission today, well, it's not just a survey. We're aiming for complete intellectual mastery.
That's right.
Going deep, really deep, right down to the fundamental structural unit of animal life, the cell itself. This deep dive is designed to equip you, our listeners, with the kind of knowledge you'd expect from an elite professional in cell biology, in histology.
And we absolutely need that level of focus because we're tackling what you could argue is the most complex ecosystem on Earth.
It really is, isn't it?
Yeah. We're going to unpack the architecture, the mechanisms of, an estimated 40 trillion cells, all operating in this coordinated harmony inside the average adult human body, all based on the core source material we're digging into.
40 trillion. I mean, that number alone just tells you the cell is anything but simple.
Absolutely not. At the, the most basic level, sure, the cell is the functional unit. It's the smallest living part of the body. But for us, for this deep dive, the critical immediate distinction is between the cells that build us, complex animals, and, everything else. We're focused squarely on eukaryotic cells.
Okay, so what's the absolute defining feature? What separates our cells from, say, a bacterium?
It really comes down to internal organization, compartments. Eukaryotic cells, our cells, are defined by being enclosed by a plasma membrane, crucially having a distinct membrane-enclosed nucleus.
The control center.
Exactly. And having a cytoplasm that's just packed with these highly specialized membranous organelles, plus they've got complex non-membranous bits, molecular assemblies, and this incredibly sophisticated dynamic cytoskeleton.
And the prokaryotes. Bacteria.
They're generally smaller, often have a cell wall, but the key difference is they fundamentally lack that enclosed nucleus and that complex internal membrane system. That membrane-bound specialization, that's really the secret sauce for complex life.
Right, that internal complexity allows for this massive specialization we see across different tissues. It's still kind of mind-bending that every single one of those trillions of specialized cells, a muscle fiber built for contraction or a neuron for communication, they all start out the same way.
They do. Every single one traces back to the zygote, that single cell formed at fertilization.
The very beginning.
The very beginning. Its initial divisions produce these cells called blastomeres, and they form the inner cell mass in the early embryo. That inner cell mass, that's the potent source. It gives rise to every single tissue type in the fetus. And, that's where embryonic stem cells come from in culture, these cells that still have the potential to become pretty much anything.
But that journey from that sort of universal, all-powerful stem cell to a highly dedicated worker cell that's differentiation, how does the cell actually decide its career path? Is there like a molecular switch?
Differentiation is essentially a process of control gene expression and repression. It's the specialization process where cells start to predominantly express certain sets of genes while silencing others. And the products of those genes, the proteins, they then mediate very specific activities in the cytoplasm. And that dictates the cell's final shape, its function, and how it contributes to the tissue.
Can we actually see that specialization reflected quickly in a physical change?
Oh, often dramatically, yeah. Take a muscle cell precursor, it gets the signal to differentiate. And what happens? It elongates massively into this fiber-like shape.
Right.
Now, all animal cells have the proteins actin and myosin. They're fundamental.
Right.
But the muscle cell differentiates by basically dedicating itself almost entirely to contraction. It starts churning out and organizing these huge, intricate arrays of actin and myosin filaments just so it can efficiently convert chemical energy, ATP, into powerful, forceful mechanical work. It ramps up that basic capacity exponentially.
And that ramping up, that enhancement, leads to this incredible functional diversity. The source material makes it clear it's often not about inventing a totally new function, but taking a basic cellular ability and just making it the cell's primary hyper-efficient job.
Precisely. If you look across the functional landscape, you see these domains clearly established. Movement, that's mastered by muscle cells and related cell types. Junction formation, creating stable barriers and connections, that's the exclusive realm of epithelial cells. Then you have the cells whose whole purpose is building the environment outside the cell. The ECM synthesizers.
Like fibroblasts?
Fibroblasts, yeah, and bone cells, cartilage cells. Their job is just pumping out the components of the extracellular matrix.
And what about the really critical task of processing information, turning signals into action?
That's the specialized work of neurons and sensory cells. They're built to convert stimuli, light, sound, pressure, chemical signals into electrical impulses, action potentials.
The language of the nervous system.
Exactly. We also see huge specialization in secretion. It varies widely. You have digestive glands pouring out enzymes versus cells in the adrenal gland or testis or ovary that are dedicated steroid factories. Even just moving salts around is specialized. Think of cells in the kidney or salivary gland ducts handling massive ion transport.
We can't forget the cellular cleanup crew either.
Right, the macrophages and neutrophils. They are the dedicated janitors specializing in intracellular digestion, basically eating debris and invaders through phagocytosis. But here's, I think, a really key insight for achieving that mastery level we're aiming for. Two cells can look structurally almost identical under a microscope and yet behave radically differently. And that difference comes down purely to their environment and, critically, the specific receptors they display on their surface.
Oh, their function isn't just what's inside, it's about what signals they can actually hear from the outside.
That's a really good way to put it, yeah. Their diverse receptor arrays are like their behavioral fingerprint. Think about hormonal sensitivity. Breast fibroblasts and uterine smooth muscle cells. Well, at a basic level, they're just connective tissue and muscle cells.
Okay.
But they express specific receptors that make them exceptionally sensitive to female sex hormones. Most other fibroblasts or smooth muscle cells in the body, they lack those specific receptors, completely insensitive. So the receptor array is the critical decider of behavior, sometimes more so than the basic cell type.
Okay, so if the cell is like this complex city-state, then the plasma membrane has got to be its dynamic border wall, right? Regulating everything coming in and going out. What's this barrier actually made of?
It's a really sophisticated composite structure. Primarily, it's phospholipids, cholesterol, and proteins. And then it's kind of decorated with these oligosaccharide chains, sugars, that are covalently attached to both the lipids and the proteins.
And its job is pretty complex, too.
Oh, absolutely. It's multifaceted. It acts as a selective barrier controlling what gets through. It facilitates the transport of specific molecules. Critically, it maintains a unique constant ion concentration inside the cytoplasm, chemically and electrically different from the outside world. And it performs specific recognition and signaling functions, too.
The fundamental structure, though, that relies on the chemistry of those phospholipids.
Exactly. Phospholipids are amphipathic. That's the key term. They have these hydrophilic, water-loving polar heads, often charged, and then these hydrophobic, water-hating, non-polar fatty acid tails.
Right. Oil and water don't mix.
Precisely. So when you put them in water, they spontaneously arrange themselves into the most stable configuration, the bilayer. The hydrophobic tails bury themselves inside, away from the water, forming this protected core, while the hydrophilic heads face outwards, interacting with the water inside and outside the cell. It's incredibly stable.
And cholesterol, which is this bulky lipid, where does that fit into this fluid sheet?
Cholesterol is the essential modulator, the stabilizer. It's a sterile lipid, and it wedges itself in among those fatty acid tails. By doing that, it physically restricts the movement of the tails. It acts almost like a scaffolding, reducing the overall fluidity, making the membrane less leaky, but also preventing it from becoming too rigid at lower temperatures. It's crucial for maintaining the right consistency.
And we also need to remember this membrane isn't symmetrical, right? It's not a perfect mirror image on both sides.
No, that asymmetry is absolutely fundamental to its function. The types of phospholipids aren't distributed evenly. For example, in red blood cells, you find specific ones like phosphatidylcholine, mostly on the outer layer, while others, like phosphatidylserine, are predominantly on the inner cytoplasmic face.
Okay.
And then layered on top of that outer surface, you have the glycocalyx. This is a kind of fuzzy coat made from the carbohydrate chains of glycolipids and glycoproteins sticking out from the membrane. It's delicate, but really important for things like cell recognition, adhesion, and even defining the cell's antigenic properties, how the immune system sees it.
So when scientists are looking at this under a powerful electron microscope, what's the visual evidence that confirms this bilayer idea?
The classic trilaminar appearance. When you fix the tissue, especially using osmium tetroxide, the osmium binds really strongly to those charged hydrophilic polar heads on both the inner and outer surfaces.
So you see two dark lines.
Exactly. Two distinct dark lines. And they sandwich this lighter band in the middle. That light band is the hydrophobic core, the fatty acid tails, where the osmium can't bind. And seeing that pattern, which is incredibly thin, only about 7.5 to 10 nanometers thick, is the direct visual confirmation of the lipid bilayer model.
Okay, let's switch to the proteins. They make up roughly half the membrane's weight. You mentioned they're sort of permanent residents and more temporary guests.
That's a good way to think about it. The permanent residents are the integral proteins. They are embedded directly within the lipid core, anchored by hydrophobic interactions with the fatty acid tails. Many of these are multi-pass proteins, meaning they snake back and forth across the membrane multiple times.
Hard to get out, presumably.
Very hard. You need strong detergents to disrupt the membrane and extract them. Then you have the temporary guests, the peripheral proteins. They're bound more loosely to one surface, usually the inner cytoplasmic side, mainly through weaker ionic bonds. So you can often wash them off just using salt solutions.
This brings us back to the famous fluid mosaic model. It describes proteins floating like icebergs in a sea of lipids. But you mentioned earlier, this movement isn't totally free-for-all.
No, it's definitely dynamic, but it's also highly regulated and tethered. Lateral movement can be restricted. In epithelial cells, for example, tight junctions act like fences, corralling proteins into specific domains, keeping apical proteins on the top surface and basolateral proteins on the bottom and sides.
Creating specialized zones.
Exactly. Movement is also restricted by direct attachments to the internal cytoskeleton, the cell's internal scaffolding.
And even within that fluid mosaic, certain lipids and proteins seem to cluster together, forming functional little neighborhoods.
Yes, those are the fascinating lipid rafts. These are specialized microdomains within the membrane. They tend to have higher concentrations of cholesterol and specific types of lipids, like saturated fatty acids, which makes them slightly less fluid, more ordered.
Oh, they're like platforms.
Kind of, yeah. They act as staging areas. They gather specific scaffold proteins, enzymes, and signaling molecules together. This allows these components to interact much more efficiently because they don't have to randomly bump into each other across the entire vast surface of the membrane. It concentrates the machinery.
Which implies this boundary, the membrane, is constantly talking to the inside of the cell. How did that physical link work?
A key player there is a class of integral proteins called integrins. Integrins are crucial because they physically bridge the gap. They link the internal cytoskeleton, specifically actin filaments, to components outside the cell in the extracellular matrix, ECM.
There's a two-way street.
Absolutely. It creates this continuous pathway for bidirectional influence. What happens outside the cell can directly influence the tension and organization of the cytoskeleton inside, and changes inside the cell can affect how it groups the matrix outside. It's a constant dialogue.
Okay, so the ultimate job of this barrier is traffic control. If a molecule needs to cross, you mentioned general mechanisms, basically categorized by whether they cost energy. Let's start with the free rides, the passive transport going downhill.
Right. Passive transport doesn't require the cell to spend any ATP. Substances just move down their concentration gradient from high concentration to low. The simplest form is simple diffusion. This works for small, non-polar molecules, things that are lipid-soluble, lipophilic. They can just slip right through the lipid bilayer.
Like oxygen, CO2.
Exactly. Gases, steroid hormones, water can too, but surprisingly slowly on its own through the lipids.
Then we get to the mechanisms that need some help. Protein assistance, but still don't cost ATP.
Correct. First up are the channels. Those are usually multi-pass proteins that form selective water-filled pores right through the membrane. Think of aquaporins, which create dedicated channels just for water to move through much faster. Or you have ion channels specific for sodium Na+, potassium K+, calcium Ca2+, and so on. These are often gated, meaning they can open and close very rapidly in response to specific signals, like a change in voltage across the membrane or the binding of a chemical ligand.
Okay, so channels are like selective tunnels. What's the other passive helper?
Those are the carriers or transporters. These are also transmembrane proteins, but instead of forming a pore, they physically bind to the specific small molecule they transport. Then they undergo a conformational change, a shapeshift, that effectively flips the molecule across the membrane. This is called facilitated diffusion. Still passive, still down the gradient.
So if that's all passive downhill travel, then the energy-intensive process must be moving things uphill against their concentration gradient. That's active transport.
Precisely. Active processes absolutely require energy, usually from ATP hydrolysis, because they're forcing substances to move against their natural pendency, against their concentration gradient, or maybe against an electrical gradient too.
And the main players here are pumps.
Yes, the primary mechanism involves pumps, which are often called ATPases because they are enzymes that directly hydrolyze ATP to power the movement. The classic examples everyone learns are the Ca2 plus pump, which keeps cytoplasmic calcium levels incredibly low, and the crucial Na plus K plus pump, which maintains the high potassium low sodium environment inside most animal cells. That's primary active transport.
Okay, so that Na plus K plus pump works constantly, burning ATP to build up this really steep sodium gradient. Lots of sodium outside, very little inside. Can the cell then leverage that stored energy?
Yes, that's the clever bit. That's secondary active transport. It's like using the energy stored in water flowing downhill behind a dam to power something else. The cell harnesses the potential energy released when sodium flows down its steep concentration gradient, which was established by the primary pump, to simultaneously drive the uphill movement of a second substance.
Okay, so sodium pays the toll for something else.
Exactly. And we see two main flavors of this. Symport, where both substances, the sodium moving downhill and the other substance moving uphill, travel in the same direction across the membrane. This is vital for absorbing nutrients, like the Na plus glucose symporter in the gut.
And the other.
Antiport, where they move in opposite directions. Sodium flows in, down its gradient, powering the export of another substance, like protons, H+, out of the cell against its gradient. The Na plus H plus antiporter is essential for regulating intracellular pH, for instance.
Okay, pumps and channels handle the small stuff. But what if the cell needs to bring in something big, like a whole protein or a chunk of fluid, maybe even engulf a bacterium and needs a bulk mechanism, right? That sounds like endocytosis.
Exactly. Endocytosis is the general term for uptake processes, where the plasma membrane folds inward, surrounds some extracellular material, and then pinches off to form a vesicle or vacuole inside the cytoplasm. And there are a few specialized types for different cargo sizes and specificities.
Let's start with the most dramatic, the cell eating.
That's phagocytosis. This is specifically for ingesting large particles, things like bacteria, dead cells, or other debris. It's mainly carried out by specialized cells like macrophages and neutrophils in the immune system. They extend these projections, called pseudopodia, driven by the cytoskeleton, to surround and engulf the particle,
Forming a little bubble inside.
Right. The resulting internal vesicle is called a phagosome. And crucially, that phagosome then has to fuse with a lysosome, the cell's digestive organelle, to break down the contents.
Then there's the less dramatic, more routine cell drinking.
That's penocytosis. It involves much smaller, often non-specific invaginations of the membrane that just capture a tiny gulp of the surrounding extracellular fluid along with any dissolved solutes. The vesicles formed are pretty small, maybe around 80 nanometers.
And what happens to them?
Usually, they also fuse with lysosomes to digest the contents, but interestingly, in some polarized cells, like the endothelial cells lining blood vessels, these penocytotic vesicles might travel right across the cell from one side to the other and release their contents on the opposite side. That process is called transcytosis, and it's a major way to move substances in bulk across cellular barriers.
Okay, so phagocytosis is for big particles, penocytosis is sort of general fluid sipping. But what if the cell needs to import something specific, like, say, all the LDL cholesterol it needs? It can't just rely on random sipping.
No, for specific cargo, it uses receptor-mediated endocytosis. This is highly selective. It relies on integral membrane proteins receptors that bind specific molecules, the ligands, with high affinity, like LDL receptors binding LDL particles.
And that binding triggers the uptake.
Yes. Ligand binding causes these receptors to cluster together in specialized regions of the membrane called coated pits. These pits are lined on the cytoplasmic side by a protein scaffold, most famously involving the protein clathrin, along with adapter proteins. This protein coat actually helps deform the membrane, driving the invagination and pinching off to form a coated vesicle containing the receptors and their bound cargo.
The source material also mentions similar little pits or invaginations that don't use clathrin.
Yes, the caveoli, meaning little caves. These are another type of specialized invagination, often found in very thin cells like endothelial cells or muscle cells. They involve different scaffolding proteins, particularly the integral protein caveolin and associated proteins called cavins. They seem to be involved in both specific types of endocytosis and also in organizing certain signaling pathways at the membrane.
So once that vesicle clathrin-coated or caveolar pinches off and gets inside, it's on a critical journey. It needs to be directed to the right place immediately, right?
Absolutely. It enters what's called the endosomal compartment. You can think of this as the cell's central sorting station, usually a network of tubules and larger vesicles, or vacuoles, located near the cell periphery initially.
First order of business.
Get rid of the coat. If it was a clathrin-coated vesicle, the clathrin coat rapidly disassembles, and the clathrin molecules are recycled back to the plasma membrane for reuse. Then the vesicle itself, now uncoated, fuses with components of the endosomal network.
And navigating this network isn't random.
Not at all. Vesicle trafficking within the endosomal system, and indeed throughout the cell, is highly directed. It relies heavily on small G proteins called Rab proteins. These act like molecular switches or address labels, ensuring that a vesicle fuses only with the correct target membrane. Different Rabs mark different pathways.
And within this sorting station, the fate of the cargo gets decided. What determines if, say, the receptor gets reused and the ligand gets destroyed?
A key factor is often pH. As vesicles mature within the endosomal system, moving towards becoming late endosomes, they acquire ATP-driven proton pumps in their membrane. These pumps acidify the interior lumen.
Okay.
This drop in pH does two critical things. First, it helps activate hydrolytic enzymes that might be present, starting the breakdown process. Second, the acidic environment often causes the ligand to detach from its receptor.
Ah, so they uncouple.
Exactly. Once unbound, the receptors can be sorted into different vesicles, often forming recycling endosomes, which ferry them back to the plasma membrane for another round of uptake. This is crucial for receptors like the LDL receptor. The ligands, now free, typically proceed towards fusion with lysosomes for complete degradation. Or in the case of transcytosis, they might be repackaged and sent to the other side of the cell.
Okay, so that's uptake. The flip side is getting stuff out exocytosis, releasing bulk material.
Right. Exocytosis is how cells secrete large molecules or waste products. A vesicle, originating usually from the Golgi apparatus, travels to the plasma membrane, fuses with it, and releases its contents to the outside. Importantly, this happens without breaking the integrity of the plasma membrane itself.
Is it always happening, or is it triggered?
Both. We distinguish between two main pathways, and often a transient rise in intracellular calcium ions acts as the trigger. Constitutive secretion is the continuous, ongoing release of substances. For example, fibroblasts constantly secrete collagen subunits to build the extracellular matrix.
Just baseline activity.
Exactly. Then there's regulated secretion. This is for substances that are produced and stored, often in high concentrations, within secretory vesicles or granules. These vesicles wait near the membrane until the cell receives a specific signal, like a hormone or neurotransmitter binding, which then triggers their fusion and release. Think of pancreatic cells releasing digestive enzymes only after a meal, or nerve cells releasing neurotransmitters.
This constant budding off and fusing back endocytosis and exocytosis means the membrane itself is in constant flux.
Absolutely. That's called membrane trafficking. Endocytosis removes patches of membrane, while exocytosis adds them back. The cell has to carefully balance these processes to maintain its overall surface area and membrane composition. And we should also briefly mention exosomes. These are very small vesicles, less than 120 nanometers, that are formed in a more complex way involving multivesicular bodies fusing with the plasma membrane. They're released by many cell types and act as tiny communication packets, transferring proteins, lipids, and even RNA between cells.
Beyond direct contact, like gap junctions, multicellular life depends on signals traveling, sometimes long distances. How does the cell handle these complex conversations?
Well, cells use families of receptors to detect these extracellular signals or stimuli, and we can categorize the communication based on how far the signal travels. Endocrine signaling is long-distance hormones are carried by the bloodstream to target cells far away.
Like insulin from the pancreas affecting muscle cells.
Perfect example. Then there's paracrine signaling, which is local. The signaling molecule diffuses only a short distance and is often quickly broken down or captured, so it only affects immediate neighbors. Synaptic signaling, at nerve junctions, is really a specialized, very fast form of paracrine signaling across a tiny gap.
Can cells talk to themselves?
They can. That's autocrine signaling. A cell releases a signal that then binds to receptors on its own surface. This can be important for reinforcing a cell's differentiation state, for example. And finally, there's juxtacrine signaling, which requires direct physical contact. A molecule embedded in the membrane of one cell binds to a receptor on an adjacent cell.
Okay, so for signals that are water-soluble, hydrophilic, they can't cross the lipid membrane barrier. They must be detected at the surface. What are the main types of surface receptors they use?
There are three major classes for these ligands, which are often polypeptide hormones or neurotransmitters, the first messengers. First, channel-linked receptors. Binding of the ligand directly opens an ion channel that's part of the receptor itself, causing a rapid influx or efflux of ions.
Very direct.
Very direct, very fast. Second, enzymatic receptors. Here, ligand binding activates an enzymatic activity inherent to the cytoplasmic part of the receptor itself. Often, this activity is a protein kinase, meaning it adds phosphate groups to other proteins.
And the third type seems like the most common and maybe most complex.
That would be the G protein-coupled receptors, or GPCRs. These are incredibly numerous and versatile. When the ligand binds to the external part of the GPCR, it causes a conformational change that allows the receptor to interact with and activate a separate protein complex on the inner membrane surface called a G protein.
Okay.
This G protein then binds GTP, hence the name, and splits into subunits that can then go on to activate or inhibit other target proteins in the cell. These might be ion channels or, crucially, enzymes like adenyl cyclase.
So it's that initial binding event, the first messenger, just triggers the first step. This then kicks off an internal signal transduction cascade, which really amplifies the signal. Right?
Amplification is absolutely key. A single receptor binding one molecule can lead to the activation of many G proteins, each of which might activate many enzyme molecules. For GPCRs involving adenyl cyclase, the activated enzyme then generates large quantities of a small intracellular signaling molecule called cyclic AMP or key MMP. This is a classic second messenger.
So the signal crosses the membrane indirectly.
Exactly. Other important second messengers generated by different pathways include diacylglycerol, DAG, and inositol triphosphate, IP3. These second messengers then diffuse through the cytoplasm, activating downstream enzymes, typically protein kinases again. These kinases phosphorylate yet other proteins, leading to a cascade effect that ultimately changes the cell's behavior, maybe by altering gene expression or activating specific metabolic pathways. The amplification can be enormous.
But not all signals need a surface receptor. What about things like steroid hormones, which are fatty, lipophilic?
Right. Hydrophobic signaling molecules, like steroids or thyroid hormones, are lipid-soluble enough that they can simply diffuse directly through the plasma membrane by simple diffusion. They don't need a surface receptor.
So where do they bind?
They bind to specific receptor proteins that are located inside the cell, often initially in the cytoplasm, or sometimes directly in the nucleus. Once the hormone binds, the activated receptor hormone complex typically moves into the nucleus, binds to specific regulatory regions on the DNA, and directly influences the rate of transcription of target genes, usually increasing it.
Okay, we've handled the border control and the communication systems. Now let's dive into the city itself, the cytosol. This is the jelly-like fluid inside, right? Holding all the machinery.
Exactly. It's this viscous, aqueous fluid filling the cell, packed with enzymes, like all the enzymes for the glycolytic pathway, and it's the medium through which substrates diffuse. And suspended within it are the organelles and the non-membranous structures. Let's start with a key non-membranous player, the ribosomes.
The protein factories.
The universal protein factories, they're these complex macromolecular machines made of ribosomal RNA, or RNA, and proteins. Their job is to assemble polypeptides, stringing amino acids together according to the instructions coded in messenger RNA, mRNA, with the help of transfer RNA, tRNA.
And they're visible in histology.
Well, areas rich in ribosomes are the RNA backbone has lots of phosphate groups, which are negatively charged, so regions of the cytoplasm that are actively synthesizing proteins and have lots of ribosomes, especially clusters called polysomes or polyribosomes.
Multiple ribosomes on one mRNA.
Right, translating the same message simultaneously. These regions stain intensely with basic dyes, which are positively charged. This staining characteristic is called basophilia, and it's a classic sign of high protein synthesis activity.
And where the ribosome actually does its work determines the protein's ultimate fate. There's a sorting decision right at the beginning.
Absolutely fundamental. If a protein is destined to function within the cytosol itself, or if it needs to be imported into organelles like mitochondria, peroxisomes, or the nucleus after it's made, then it's synthesized on free polyribosomes, ribosomes just floating freely in the cytosol.
Okay.
However, if the protein is meant to be secreted out of the cell or embedded within a membrane, plasma membrane, ER, Golgi, lysosome, or packaged into a lysosome, then its synthesis must occur on polyribosomes that become attached to the surface of the endoplasmic reticulum, ER.
Which brings us to the RER, the rough endoplasmic reticulum, called rough because of those attached ribosomes. It's prominent in cells that are secretion powerhouses,
Right.
like pancreatic cells making digestive enzymes, or plasma cells making antibodies.
Exactly. The RER is this extensive network of interconnected, flattened, membrane-bound sacs called cisternae. It's often directly continuous with the outer membrane of the nuclear envelope. Its surface is studded with those ribosomes actively synthesizing proteins destined for the secretory pathway or membranes.
And its job isn't just synthesis, it's also about processing and quality control.
Crucially, yes. As proteins are synthesized and threaded into the RER lumen, the space inside the cisternae, several things happen. They undergo initial modifications, like the addition of core sugar chains, glycosylation. They start to fold into their correct three-dimensional shapes, often assisted by other proteins inside the RER called molecular chaperones. Multi-subunit proteins begin to assemble, and this whole process is under strict quality control surveillance by those chaperones.
So how does a ribosome, which starts translating out in the cytosol, know it's supposed to go to the RER instead of just finishing the protein free? Is there a signal?
There is a very specific signal. It's encoded right at the beginning of the mRNA sequence for these particular proteins. As the ribosome starts translating, the first part of the polypeptide chain to emerge is the N-terminal signal sequence, typically 1540 amino acids long, often hydrophobic.
Like a zip code.
Precisely. As this signal sequence emerges, it's immediately recognized and bound by a cytosolic complex called the signal recognition particle, or SRP. Binding of the SRP acts like a temporary brake, pausing further translation.
Okay, pauses the factory. Then what?
The SRP then acts as a guide. The entire complex SRP ribosome, partially made polypeptide, and mRNA diffuses to the RER membrane, where the SRP binds to a specific SRP receptor. This docking event brings the ribosome right next to a protein channel in the ER membrane called the translocon.
The entry gate.
The entry gate. The SRP dissociates, translation resumes, and the growing polypeptide chain is threaded through the translocon into the RER lumen. And usually, very early on, an enzyme called signal peptidase located inside the lumen cleaves off that signal sequence. Its job is done. The protein is inside. Translation continues until the protein is fully synthesized within the RER lumen, where chaperones help it fold correctly.
That quality control by the chaperone sounds incredibly important. What happens if a protein just doesn't fold right?
It's a life or death check for the cell. If a protein is terminally misfolded, if the chaperones just can't get it into the right shape, it's flagged as unsalvageable. It's then targeted for destruction through a process called ER-associated degradation, or ENAD.
How does that work?
The faulty protein is actually recognized and physically translocated back out of the RER through a channel into the cytosol. Once in the cytosol, it gets tagged with a chain of small proteins called ubiquitin, marking it for destruction by the cell's primary protein disposal machine, the proteasome.
Okay, so the proteasome is this non-membranous machine, distinct from the lysosomes, which handle bigger things like whole organelles. The proteasome deals with individual faulty molecules.
That's the critical distinction, yes. Proteasomes are these barrel-shaped, highly abundant protein complexes found throughout the cytosol and nucleus. Their specific job is to degrade proteins that are denatured, misfolded, or perhaps correctly folded but just naturally short-lived, like regulatory proteins whose levels need to change quickly.
How does the cell ensure only the intended targets get shredded? It seems risky.
It uses that universal tag for destruction we just mentioned, ubiquitin. The protein targeted for degradation must first be polyubiquitinated, meaning multiple ubiquitin molecules are covalently attached to it, forming a chain.
So the ubiquitin chain is the signal.
It's the signal. The proteasome itself has regulatory cap particles at each end of its cylindrical core. These caps recognize the polyubiquitin chain, bind the tagged protein, unfold it, which requires ATP energy, and then thread it into the central core of the proteasome. Inside the core are active protease enzymes that chop the protein up into small peptides.
This system sounds absolutely essential for preventing, well, cellular chaos from accumulating junk proteins.
It is fundamental housekeeping. And failures in this quality control system, either in ERAD identifying faulty proteins or in the proteasome itself, can have devastating consequences. The accumulation of large insoluble aggregates of misfolded proteols within and around cells is now recognized as a central pathological mechanism in major neurodegenerative diseases, most notably Alzheimer's disease and Huntington's disease.
Okay, next to the RER, we often find the SER, the smooth endoplasmic reticulum. Smooth because it lacks ribosomes. It's often more tubular or sac-like. Since it's not directly making proteins, what are its main jobs, which can vary by cell type?
The SCR has several key diverse functions. First, it's a major site for lipid synthesis. Enzymes embedded in the SCR membrane synthesize phospholipids for all the cell's membranes and also cholesterol. In cells that specialize in making steroid hormones, like those in the adrenal cortex or gonads, the SCR is incredibly prominent because steroids are derived from cholesterol.
Okay, lipid factory. What else?
Second major role, detoxification. Especially in liver cells, the SCR membrane is packed with enzymes, particularly from the cytochrome P450 family. These enzymes chemically modify and detoxify a huge range of potentially harmful substances, both exogenous compounds like drugs and alcohol, and endogenous molecules like bilirubin or components of bile. This makes the SCR exceptionally abundant in hepatocytes.
And a third major function related to rapid signaling inside the cell.
Yes, that's calcium sequestration and release. The SCR acts as an internal reservoir, actively pumping calcium ions from the cytosol into its lumen and storing them. Then, in response to specific signals, it can rapidly release that stored calcium back into the cytosol. This calcium spike acts as a crucial intracellular signal, triggering many rapid responses, like muscle contraction. In fact, in striated muscle cells, this specialized calcium-storing SCR is so important it gets its own name, the sarcoplasmic reticulum.
And the source material notes a really practical clinical connection here, especially concerning newborns.
Yes. A common, usually temporary form of jaundice seen in newborns happens because the SCR in their liver cells is still underdeveloped right after birth. It hasn't reached full functional capacity yet, so it can't efficiently process bilirubin, which is a yellow breakdown product of old red blood cells, into a form that can be easily excreted by the body. Bilirubin builds up, causing the yellowish skin and eyes. It usually resolves as the liver matures.
Okay, moving along the production line. Proteins and lipids made in the ER, especially the RER, are then often shuttled to the cell's central processing, sorting, and packaging hub, the Golgi apparatus, or Golgi complex.
Right. The Golgi is this dynamic stack of flattened, smooth, membrane-bound sacs called cisternae, typically located near the nucleus and the ER. You can think of it as the cellular post office. It receives newly synthesized proteins and lipids from the ER, further modifies and sorts them, and then packages them into vesicles for delivery to their final destinations.
And there's a clear direction of flow.
Yes, it's highly organized. Transport vesicles bud off the RER and fuse with the first compartment of the Golgi stack, known as the cis-face or cis-Golgi network, CGN. That's the receiving side. From there, the cargo moves sequentially through the stack, through the medial cisternae, where most of the enzymatic modifications happen. Finally, processed molecules reach the transface, or trans-Golgi network, TGN, the shipping side, where they are sorted and packaged into different types of vesicles that bud off.
How does the cell keep the vesicles moving in the right direction within the Golgi itself and ensure they fuse correctly?
It uses specific protein coats on the budding vesicles and those Rab proteins, again, for targeting. Generally, COP2-coated vesicles mediate the forward anterograde movement from the ER to the cis-Golgi. Copy-coated vesicles are primarily involved in retrograde movement, retrieving escaped ER-resident proteins or recycling membrane components back from the Golgi to the ER. And as always, specific Rab proteins on the vesicles and target membranes ensure they fuse with the correct compartment.
What kind of critical chemical modifications are finalized in the Golgi that the RER only started?
The Golgi is where the really complex post-translational modifications happen. It elaborates on the simple core glycosylation that occurred in the RER, adding more complex sugar chains. It can also perform sulfation, adding sulfate groups, phosphorylation, adding phosphate groups, and even some limited proteolytic cleavage, cutting proteins into their final active forms. And critically, it concentrates, sorts, and packages products for secretion or delivery to other organelles like lysosomes. The concentration step can be significant products destined for regulated secretion might be concentrated up to 200 times compared to their concentration in the RER.
And when those highly concentrated products are ready for storage, awaiting a signal for release, they form specific packages.
Yes, they bud off the transface packaged into secretory granules. If these granules contain inactive enzyme precursors, like in the pancreas, they're often called zymogen granules. These granules are essentially stored packets of concentrated product held in the cytoplasm near the plasma membrane, ready for rapid, regulated exocytosis when the right signal arrives.
Okay, now let's turn to the cell's internal demolition and recycling center, the lysosomes. They handle breakdown of waste and cellular turnover.
Right. Lysosomes are membrane-bound vesicles filled with a potent cocktail of about 40 different acid hydrolase enzymes. These include proteases, nucleases, lipases, glycosidases enzymes capable of breaking down virtually all types of biological macromolecules. The key feature is that these enzymes work optimally at an acidic pH, around 5.0.
Which is much lower than the cytosol's pH. How is that maintained?
The lysosomal membrane contains ATP-driven protar pumps that actively pump H+ ions from the cytosol into the lysosome lumen, maintaining that acidic environment. This serves as a safety mechanism, too. If a lysosome were to leak, the enzymes wouldn't be very active in the neutral pH of the cytosol.
The cell has to get these dangerous enzymes safely into the lysosome in the first place, without them digesting things on the way. How does it target them specifically during their journey through the Golgi?
This is a beautiful example of molecular sorting. The lysosomal hydrolyses are synthesized in the RER, like other secretory proteins, and travel into the Golgi. Within the cis-Golgi network, these specific enzymes are recognized, and a unique molecular tag is added to them. Mannose 6-phosphate, M6P.
M6T, that's the lysosome address label.
Exactly. Then, in the trans-Golgi network, there are specific M6P receptors embedded in the membrane. These receptors bind tightly to the M6P-tagged enzymes, effectively capturing them and diverting them away from the vesicles destined for secretion. Instead, they are packaged into clathrin-coated vesicles that bud off and eventually mature into or fuse with lysosomes. These newly formed vesicles containing only the enzymes are sometimes called primary lysosomes.
That M6P system sounds incredibly precise, but also like a potential point of failure. What happens if that tagging system messes up? Is there a backup?
It is a major vulnerability. If the enzymes responsible for adding the M6P tag are defective, the lysosomal hydrolases aren't recognized by the receptors in the TGN. Instead, they get mistakenly packaged into secretory vesicles and secreted constitutively out of the cell.
So the lysosomes end up empty.
Essentially empty of their digestive enzymes, yes. This leads to a devastating group of genetic disorders called lysosomal storage diseases. In eye cell disease, for example, caused by a defect in the M6P tagging enzyme, multiple types of undigested macromolecules accumulate inside lysosomes in various tissues, causing severe cellular dysfunction and developmental problems. There's really no effective backup system for this targeted delivery.
So assuming the enzymes get there correctly, a primary lysosome then fuses with a vesicle containing material brought in by endocytosis, like a phagosome. What's the resulting structure called?
That fusion creates a secondary lysosome, sometimes called a heterolysosome. Now you have the digestive enzymes mixed with the material to be digested. The proton pumps ensure the pH drops, the hydrolases become fully active, and digestion begins.
And lysosomes don't just digest stuff from outside. They also clean up the cell's own internal components, right?
Yes, that vital process is called autophagy, meaning self-eating. When organelles like mitochondria become damaged or old, or if the cell needs to recycle components during starvation, a membrane, often originating from the ER, envelops the target organelle or chunk of cytoplasm, forming a double membrane vesicle called an autophagosome.
Okay.
This autophagosome then fuses with a lysosome, delivering its contents for degradation. It's a crucial mechanism for maintaining cellular health and quality control and for survival under stress.
What happens to the stuff the lysosome just can't break down, the indigestible leftovers?
Any undigestible material remains within a membrane-bound vacuole called a residual body. In cells that don't divide or divide very slowly, long-lived cells like neurons or cardiac muscle cells, these residual bodies can accumulate over the lifespan of the organism. They often contain oxidized lipids and proteins, forming a brownish granular pigment called lipofuscin, sometimes referred to as the age pigment.
And other lysosomal storage diseases, besides eye cell, happen when just one specific enzyme is missing.
Yes, many lysosomal storage disorders like Tay-Sachs disease or Gaucher disease are caused by a genetic defect in a single specific lysosomal hydrolase. This leads to the accumulation of the particular substance that enzyme normally degrades, causing specific cellular and clinical problems depending on the enzyme and the tissue affected.
All right, let's move to the undisputed energy engine of the cell, the mitochondria. These are the primary sites of ATP synthesis through aerobic respiration, absolutely essential for meeting the huge energy demands of those trillions of cells we started with.
Indeed. Mitochondria take fuel molecules, primarily pyruvate from glucose breakdown in fatty acids, and oxidize them completely to carbon dioxide and water. In doing so, they capture a huge amount of energy to produce ATP, yielding roughly 15 times more ATP per glucose molecule than anaerobic glycolysis alone. They are the powerhouses.
And they're dynamic, not just static beans.
Highly dynamic and plastic. They constantly move, fuse together, and divide fission. Their shape can vary, often appearing as elongated organelles, maybe 0.5 to 1 micrometer in diameter, and their number within a cell directly reflects the cell's energy requirements. They're incredibly abundant in cells with high energy demands, like heart muscle cells or kidney tubule cells that do a lot of active transport.
Structurally, they're famous for having two membranes.
Yes, a double envelope. The outer membrane is relatively smooth and contains numerous channel proteins called porins. These porins make the outer membrane quite permeable to small molecules and ions, less than about 5,000 Daltons, allowing them easy access into the space between the membranes, the inner membrane space.
But the inner membrane is different.
Very different. The inner membrane is the real barrier and the site of ATP synthesis. It's extensively folded into shelf-like or finger-like projections called cristae, which dramatically increase its surface area. Unlike the outer membrane, the inner membrane is highly impermeable to most ions and small molecules. It contains specific transport proteins to control what crosses it, and critically, it houses the protein complexes of the electron transport chain and ATP synthase.
And the space enclosed by the inner membrane.
That's the innermost compartment, the matrix. The matrix is packed with enzymes, including those for the citric acid cycle, also called the Krebs cycle, which further oxidizes fuel molecules derived from pyruvate, and the enzymes for the beta oxidation of fatty acids. These processes generate high-energy electron carriers, primarily NADH and FADH2, which then donate their electrons to the electron transport chain in the inner membrane.
Okay, so how does the mitochondrion actually convert the energy from those electron carriers, like NADH, into the usable energy currency, ATP?
This happens through a process called oxidative phosphorylation, driven by what's known as the chemiosmotic process. It's one of the most elegant mechanisms in biology. The electron transport chain consists of several large protein complexes embedded in the inner mitochondrial membrane. Electrons from NADH and FADH2 are passed sequentially down this chain from one complex to the next in a series of oxidation-reduction reactions, ultimately being transferred to oxygen, which is reduced to form water. Crucially, as the electrons move down the chain, several of these complexes use the energy released to actively pump protons, H+ ions, from the mitochondrial matrix across the inner membrane into the intermembrane space.
So they're building up a proton stockpile in that space.
Exactly. They create a steep electrochemical gradient across the inner membrane, both a concentration difference, pH gradient, and an electrical potential difference, membrane potential. This proton gradient represents stored potential energy, often called the proton motive force.
Like water building up behind a dam.
That's the classic analogy, yes. And just like water flowing through a turbine in a dam generates electricity, the mitochondrion harnesses a flow of protons back down their electrochemical gradient from the intermembrane space back into the matrix to generate ATP.
Through a specific machine.
Through a remarkable molecular machine called ATP synthase. This large enzyme complex is also embedded in the inner membrane and has a channel that allows protons to flow back into the matrix. This flow of protons drives a mechanical rotation within parts of the ATP synthase complex. It's like a tiny molecular motor. And this rotation is mechanically coupled to the enzymatic site that catalyzes the synthesis of ATP from ADP and inorganic phosphate. It literally converts the energy of proton flow into the chemical bond energy of ATP, churning out over 100 ATP molecules per second.
Wow. Mitochondria also have a darker role, though, in deciding cell fate.
They do. Under conditions of severe cellular stress or damage, mitochondria can be induced to initiate programmed cell death or apoptosis. A key step involves the release of certain proteins normally sequestered within the intermembrane space, most notably cytochrome C, into the cytoplasm. Cytochrome C then activates a cascade of cytoplasmic proteases called caspases, which systematically dismantle the cell in a controlled, non-inflammatory way.
And finally, the thing that makes mitochondria unique among organelles, their autonomy, which points to their evolutionary history.
Yes, the endosymbiotic theory. Mitochondria are semi-autonomous. They arise only by the fission-division of pre-existing mitochondria, and uniquely within their matrix, they contain their own small circular DNA molecule, mtDNA, as well as their own ribosomes, messenger RNAs, and transfer RNAs, all distinct from the nuclear DNA and cytoplasmic machinery, and actually more similar to those found in bacteria.
So they were likely once free-living bacteria.
That's the widely accepted theory. Billions of years ago, an ancestral eukaryotic cell likely engulfed an aerobic prokaryote, a bacterium capable of efficient respiration. Instead of being digested, this bacterium established a symbiotic relationship, eventually evolving into the mitochondrion, providing ATP in exchange for shelter and nutrients. This ancient origin is clinically relevant to certain diseases, like MRF, myoclonic epilepsy with ragged red fibers, are caused by mutations specifically in the mitochondrial DNA genes, affecting the synthesis of key proteins in the respiratory chain, particularly impacting muscle and nerve cells.
Okay, one more membrane-bound organelle to cover, the peroxisome. These are small, roughly spherical bodies enclosed by just a single membrane. What's their main claim to fame, judging by the name?
Peroxisomes are named for their central role in producing and degrading hydrogen peroxide, H2O2, which is a reactive and potentially toxic molecule. They contain enzymes called oxidases that remove hydrogen atoms from various organic substrates, transferring them to oxygen and producing H2O2 in the process.
But H2O2 is dangerous, right?
It is. So crucially, peroxisomes also contain vast amounts of another enzyme, catalase. Catalase very efficiently breaks down the H2O2 generated by the oxidases into harmless water and oxygen gas, so they both produce and neutralize H2O2 within the same compartment.
What kinds of reactions generate the H2O2? Are they detoxifiers?
Yes, they play a significant role in detoxification, particularly in liver and kidney cells, helping to break down toxins like alcohol and certain drugs. They also have specialized metabolic roles. For instance, they are the primary site for the beta oxidation of very long-chain fatty acids, those with 18 carbon atoms or more. Mitochondria handle shorter chains more efficiently. Peroxisomes shorten these very long chains before sending them to the mitochondria for complete breakdown. They're also involved in the synthesis of certain lipids, including cholesterol and bile acids.
And if the proteins needed to build or run the peroxisome are faulty, we see serious diseases.
Absolutely. Defects in peroxisomal proteins cause a range of severe disorders. For example, neonatal adrenoleukodystrophy, NALD, results from a faulty transporter protein needed to get those very long-chain fatty acids into the peroxisome for breakdown, leading to their accumulation and severe neurological damage. Zellweger syndrome is an even more severe condition caused by defects in the proteins required for importing all peroxisomal enzymes, essentially resulting in empty peroxisomes and profound developmental problems.
Okay, we've detailed the organelles, the factories, the power plants, the recycling centers, but none of this intricate machinery would be organized, nor could the cell move or maintain its shape without the internal scaffolding, the cytoskeleton. This isn't static scaffolding, though. It's a dynamic array of 3 distinct types of protein filaments.
Right. This internal framework is responsible for maintaining cell shape and polarity, organizing the organelles within the cytoplasm, providing mechanical strength, and driving cellular movements like crawling, contraction, and the transport of materials internally. And the 3 filament types, microtubules, microfilaments, and intermediate filaments, differ significantly in their size, structure, protein composition, dynamics, and specific functions.
Let's start with the largest ones, microtubules. These are fine, hollow tubes, about 25 nanometers in diameter.
That's right. The basic building block is a dimer protein composed of 2 related globular proteins, alpha-tubulin and beta-tubulin. These alpha-beta-tubulin heterodimers polymerize end-to-end to form linear chains called protofilaments. Typically, 13 of these protofilaments associate side by side to form the hollow wall of the microtubule.
And they're polarized. They have directionality.
Yes, very much so. Because of the way the tubulin dimers add on, the microtubule has inherent polarity. One end, designated the plus end, typically grows faster by addition of tubulin dimers. The other end, the minus end, is usually less dynamic or anchored.
The most striking feature of microtubules, though, is their ability to appear and disappear rapidly.
That's their characteristic dynamic instability. Microtubules are constantly undergoing cycles of polymerization, growth, and rapid depolymerization, shrinkage, or catastrophe. This dynamic behavior is regulated by the binding and hydrolysis of GTP by the beta-tubulin subunit.
How does GTP control it?
When tubulin dimers carrying GTP are added to the plus end, they form a stabilizing GTP cap. As long as GTP tubulin is being added faster than the GTP is hydrolyzed to GDP within the microtubule wall, the cap remains and the microtubule grows. However, if the rate of addition slows down, the GTP hydrolysis catches up. When the plus end consists of GDP-bound tubulin, the protofilaments tend to fray apart, leading to rapid and often complete disassembly of the microtubule.
And the cell doesn't just let these grow randomly. There's an organizing center.
Correct. The nucleation, initiation, and organization of most microtubules in animal cells occurs at microtubule organizing centers, or MTOCs. The primary MTOC in most animal cells is the centrosome, typically located near the nucleus. The centrosome contains a pair of cylindrical structures called centrioles, each composed of 9 sets of triplet microtubules arranged in a characteristic pinwheel pattern, surrounded by a cloud of amorphous material called pericentriolar material, which contains the gamma-tubulin ring complexes that actually nucleate microtubule growth. Microtubules typically grow outward from the centrosome, with their minus ends anchored there and their plus ends extending towards the cell periphery.
This radiating network acts like the cell's internal railway system, doesn't it, for moving cargo around?
Exactly. It provides tracks for intracellular transport. This movement is powered by ATP hydrolyzing motor proteins that bind to microtubules and walk along them, carrying attached cargo like vesicles or organelles. There are 2 main families. Kinesins typically walk towards the plus end of the microtubule, driving anterograde transport, moving things away from the cell center towards the periphery. Cytoplasmic dyneins, on the other hand, generally walk towards the minus end, mediating retrograde transport, bringing cargo back towards the cell center, towards the nucleus and centrosome.
And because microtubules are absolutely essential for forming the mitotic spindle during cell division, they become a prime target in cancer therapy.
Precisely. Many successful chemotherapy drugs like vinblastine or paclitaxel Taxol work by interfering with microtubule dynamics. Some prevent polymerization, others prevent depolymerization. Either way, they disrupt the formation and function of the mitotic spindle, thereby arresting cell division, which disproportionately affects rapidly dividing cancer cells.
Okay, moving down in size to the thinnest filaments, the microfilaments, also known as actin filaments. These are only about 5-7 nanometers thick and are key players in cell shape changes and motility.
Right. These are flexible yet strong polymers assembled from monomers of the globular protein G-actin. G-actin monomers polymerize to form a double-stranded helix called filamentous, or F-actin. Like microtubules, actin filaments are also polarized with distinct plus and minus ends. The plus end, often called the barbed end, is typically where polymerization occurs more rapidly, while the minus end, or pointed end, is often where dissociation dominates.
This difference in growth rates at the ends leads to a phenomenon called treadmilling.
Yes, treadmilling can occur under certain conditions where monomers add to the plus end at roughly the same rate they dissociate from the minus end. This results in a net flow of subunits through the filament, even if the overall length doesn't change much. It allows for rapid reorganization of actin structures. Actin filaments are highly dynamic and are especially concentrated just beneath the plasma membrane, forming the cell cortex, which provides mechanical support and drives changes in cell shape. Their assembly is often initiated by specific nucleating proteins, like formin, which make parallel bundles, or the RP23 complex, which creates branch networks essential for pushing the membrane forward during cell crawling.
And do actin filaments also have motor proteins associated with them?
They do. The primary motor proteins that interact with actin filaments belong to the myosin superfamily. Myosins use the energy of ATP hydrolysis to walk along actin filaments, typically moving towards the plus barbed end, though myosin the 6 is a notable exception that moves towards the minus end.
What kind of jobs does this actin-myosin interaction perform?
A huge range. It's the basis of muscle contraction, obviously, involving specialized myosin the 2. But in non-muscle cells, myosin motors interacting with actin networks drive essential processes like cytokinesis, the pinching in of the cell during division using a contractile ring of actin and myosin the 2, generating force for cell migration, amoeboid movement, cytoplasmic streaming, stirring the cell contents, and the formation of cell surface projections like lamellipodia, sheet-like extensions for crawling, and stable microvilli, finger-like projections that increase surface area. Myosin motors are also involved in endocytosis.
Okay, finally, the third component. Intermediate filaments. They're intermediate in size, about 8-10 nanometers, but crucially, they are the most stable of the 3 cytoskeletal elements. Their main role seems to be providing mechanical strength.
Exactly. Intermediate filaments, IFs, are less dynamic than microtubules or actin. They're built for strength and resilience, protecting cells from physical stress. They are composed of various different types of protein subunits, which first form coiled coil dimers. These dimers then associate in an anti-parallel fashion to form tetramers, and these tetramers assemble end-to-end and side-by-side into strong, rope-like or cable-like protofibrils and finally filaments. A key structural difference from the other 2 is that assembled intermediate filaments do not appear to have polarity. The ends are equivalent.
And unlike actin and tubulin, which are pretty universal, the actual protein subunits making up IFs vary greatly between different cell types. This diversity is clinically important, right?
Hugely important. The specific type of IF protein expressed is often characteristic of a particular cell lineage. Let's run through the major classes. The keratins, or cytokeratins, are a large family found exclusively in all epithelial cells. They form extensive networks, often anchoring to specialized intercellular junctions called desmosomes and hemidesmosomes, providing tremendous tensile strength to epithelial sheets, like our skin. The process of keratinization in skin is a key adaptation allowing terrestrial life. Genetic defects in keratin genes cause blistering skin disorders like epidermolysis bullosa simplex.
Okay, so keratins for epithelia. What about other cell types?
Fimentin is the most widely distributed class III IF protein, typically found in cells derived from embryonic mesenchyme, like fibroblasts and endothelial cells. Desmin is found specifically in almost all muscle cells, smooth, skeletal, cardiac, where it helps link the contractile apparatus together. GFA, glial fibrillar acidic protein, is the characteristic IF of astrocytes and some other glial cells in the central nervous system. And finally, the lamins, lamin A, B, C, are unique because they form a meshwork called the nuclear lamina just underneath the internuclear membrane in virtually all animal cells, providing structural support to the nucleus and anchoring chromatin.
What's really striking is how this tissue specificity, combined with their stability, makes IFs an indispensable tool in pathology.
It's a cornerstone of diagnostic pathology, used routinely. Because IFs are so stable and their protein composition is so tightly linked to cell type, using antibodies to detect specific IF proteins in a tumor biopsy, immunocytochemistry, can reveal the cellular origin of that tumor with high certainty. For instance, finding GFA in a brain tumor strongly indicates it's an astrocytoma. Detecting keratins confirms an epithelial origin, a carcinoma, while vimentin might suggest a sarcoma, a synchymal origin. It's incredibly powerful for diagnosis and guiding treatment.
Okay, let's wrap up our tour of the cytoplasm with inclusions. These are different from organelles, right? They're generally temporary components, often lacking metabolic activity, and usually not enclosed by a full membrane.
That's the general idea. There are often storage depots or deposits of various substances. The main exception to the no membrane rule is lipid droplets. These store neutral fats, triglycerides and cholesterol esters, but are unique in being enclosed by only a single phospholipid monolayer, not a bilayer, studded with specific proteins. They are obviously abundant in adipocytes, fat cells, but also found in steroid-producing cells like adrenal cortex and liver cells. They often appear as clear vacuoles in standard histological preparations because the lipids get dissolved out by the organic solvents used.
What about stored energy in the form of carbohydrates?
That's stored as glycogen granules. Glycogen is a large branched polymer of glucose. In electron micrographs, these granules appear as electron-dense clumps or aggregates scattered in the cytosol. They serve as a readily accessible reserve of glucose, particularly abundant in liver cells for regulating blood glucose, and muscle cells for their own energy needs.
And finally, the various pigmented deposits we might see.
Right. There are a few common ones. Melanin is the dark brown or black pigment produced by melanocytes, primarily important for protecting the skin and eyes from UV radiation damage to DNA. Lipofuscin, as we mentioned with lysosomes, is that yellowish-brown granular pigment, a complex mixture of oxidized lipids and proteins derived partly from residual bodies. It tends to accumulate progressively with age and stable, long-lived cells like neurons and heart muscle cells, the wear and tear pigment. And hemosiderin is a dense, golden-brown iron storage complex. It consists of aggregates of denatured ferritin protein, the main intracellular iron storage protein, with bound iron. It becomes prominent in phagocytic cells, especially in the liver and spleen, after they break down old red blood cells and need to store the recycled iron.
While storing iron is necessary, too much hemosiderin can be a problem.
Yes. While some hemosiderin deposition, hemosiderosis, might be harmless, excessive accumulation of iron, leading to large amounts of hemosiderin, as seen in the genetic disorder hemochromatosis or other conditions of iron overload, can be toxic and cause significant damage to tissues, particularly the liver, heart, and pancreas.
We have covered an incredible amount of ground. We've meticulously mapped this cellular universe, starting from how differentiation drives specialization from a single zygote, exploring the plasma membrane as this dynamic, selective fortress, delving into the intricate, interconnected manufacturing and shipping lines of the RER and Golgi, the quality control systems like ER and proteasomes, the power generation in mitochondria, the recycling centers of lysosomes and peroxisomes, all supported and organized by the incredibly versatile and varied cytoskeleton.
And achieving that true professional mastery we talked about really means appreciating not just the structures, the organelles, but the specific molecular mechanisms at play. Understanding the M6P tag for lysosomal targeting, the SRP cycle for RER docking, the roles of kinesin and dynein on microtubule tracks, the clinical significance of identifying specific intermediate filaments. And crucially, recognizing how a defect in almost any single one of these tightly integrated systems can lead to specific cellular dysfunction and disease. The cell truly is the sum of its interacting parts and pathways.
We spend a lot of time defining the nucleus as the central controller, holding the primary genetic blueprint, and the cytoplasm as the machinery carrying out those instructions. But we also highlighted that the mitochondria, the absolute bedrock of cellular energy, still retain their own ancient separate DNA and their own ribosomes, operating somewhat independently of that nuclear controller.
And this is where we land, with that profound echo of the endosymbiotic past. Since mitochondria carry and replicate their own circular genetic code, and we know that mutations specifically within that mitochondrial DNA can cause debilitating human diseases independent of nuclear genes. The question this leaves us with, the one for you to really ponder, is this. Just how much of our fundamental cellular behavior, our cells' ability to survive, to function, and ultimately the processes underlying our own longevity and aging is truly dictated by this ancient, independent, symbiotic genetic code residing in our mitochondria? How much power does the host nuclear genome actually exert over the very energetic foundation upon which the entire complex cellular enterprise is built?
A fascinating question. It reminds us that even at this fundamental level, the unit of life holds on to deep evolutionary secrets, still influencing our biology today.